Great. Hello and welcome everyone to our next talk from Cornelius Römer and Fabian Engelniederhammer. Cornelius studied physics at the University of Cambridge and then switched to bioinformatics and is now part of the Pathogen evolution group at the University of Basel. Fabian is a senior consultant at TNG, working as an experienced full-stack engineer and developing open source software with our client. Now, Cornelius and Fabian will present Loculus, a highly configurable, soon-to-be open source platform for sharing genomic data.
Thank you for the introduction. Good morning, everyone. Welcome to I think it's our first official presentation on this topic, on the software we've been developing, a genomics data sharing platform. So when we talk about genomics data sharing, I think the most salient reminder of the importance has been the pandemic recently. And let's start with a picture of the case numbers of SARS-CoV-2 in the UK. So what you see there is lots of peaks, right? You remember the waves. We had them in Germany. They had them in the UK. Lots of peaks. Now, I've put this picture in black because that's essentially what you would get without genomic data. You just see peaks, and you'd not really know why there are peaks at certain times. I mean, for flu, we have peaks in the winter. Here, we had peaks in summer as well. It would be mysterious. Now, if you use genomic data, the picture suddenly gets some color, right? And the colors, they are the variants you probably have heard about, the Greek letter ones, Omicron, Alpha, Delta. And what you notice is that the peaks, they were often caused by variants. So that's why, for example, we have summer waves and not just winter waves. You see that in December 2021, we had this huge spike of Omicron BA1, that purple lilac. And we knew that was coming before the peak happened because we had genomic evidence of a new variant having developed. And that's just one kind of starter of why genomic data is valuable and how it relates maybe to your everyday life or did in the past. Now, as an aside, why I'm here also giving the talk. So as part of my job, when I'm not developing the software, I am kind of in charge of assigning these more cryptic names to these variants. You might have seen them more recently. They've become kind of labels on vaccines as well, because the WHO committee that gave these Greek letters kind of stopped giving them out because the pandemic became less important. I mean, the virus didn't stop evolving. It still keeps creating these variants. It's just kind of no longer of the same importance as it was at the height of the pandemic. But for things like vaccine development, it's still very important to monitor the evolution, and so you need names for things. So that's kind of what I do. And all of this is enabled by genomic data, and you need to share that data. Now, genomic data is produced by thousands of labs across the world. So this is a snapshot, a subset of all the data that was produced just for SARS-CoV-2. One virus out of thousands of viruses in the world. There are hundreds of human pathogens, and this is just one of them. And each of these bubbles is a single division, so something like Bavaria and Germany. And for each of these bubbles, we have data about SARS-CoV-2 viruses that were in people there. And so you see, it's essentially pretty much everywhere across the world. I mean, it doesn't matter. Every country, even kind of in sort of Saharan Africa, in the depth of Siberia, in the jungle in Brazil, people have sequenced this virus in people. And that's kind of special because often, you don't get, when you deal with data, you sort of have a couple of data producers. Maybe if you deal with global data, temperature stuff, it's often satellites capturing it. In this case, you actually have people on the ground who are putting swabs in people's noses, and they go to a lab, sequence it, and then need to submit it somewhere. And you've got people on the ground everywhere around the world doing this. It's been probably billions of dollars that have been spent on, just for SARS-CoV-2 doing the surveillance. So that's really important, that you've got this very heterogeneous group of people who create the data. And at the same time, something that's changed recently is the sheer amount of data that's been produced. So you see on this graph here the number of virus sequences that have been shared publicly per year in thousands. So it was kind of 10,000 per year in 1995. Then it rose to 100,000 in 2010. So there was a kind of exponential growth. This is a log chart. So exponential growth. That was the genomics revolution when things became much cheaper and easier to sequence. From 2010 until 2020, you see the growth slowed down. And this is probably because sequencing cost is only a fraction of the cost of sequencing these viruses. It's the expensive part in many ways is also the putting swabs in people's noses, doing PCR tests, the manual labor involved. And so that doesn't get cheaper at the same rate as genomics anymore. So that's why you get the slower growth. But then suddenly in 2020, the pandemic hit, and it became really important for public health to be able to say, when is the next wave going to hit? Where is it coming from? What do we need to put in the vaccines to be optimally protected? And so that's when you get another tenfold increase in the number of sequences shared. And in this time here, there were just enough. There were few enough viruses being shared that you could kind of get by with manual processes in many places. There was, if you think about 200,000 sequences per year, that's about 1,000 per day. So that's kind of the number of things that a human or a couple of humans can review. But when you go to 10 times that, it kind of becomes impossible. I mean, because you get data from all over the world, things like, human error happens all the time. People will put the wrong date, we see it every year that, there's a new year, and people put the collection date, they've got it hard-coded for the year before. And so things that kind of worked in the past no longer work in the same way when you have 10 times the amount of data. That's kind of the another aspect of, why we're developing the software now. And the software we're developing, it's kind of this hub in the middle. You've got on the left side the data producers. They involve, swabbing people across the world, sequencing the data. They need to share the data. I mean, that data is much more valuable if it's shared rapidly with people, so they can look which variants are going to come up, or scientists can collaborate on publications. And on the other side, you have users who, again, are heterogeneous. I mean, these data have a lot of value. You can do a lot of analyses with these. And it becomes much more valuable when you have a lot of data in one place in a standardized fashion. If you have to if every group, if every lab had its own format of sharing the genomic data, you'd get nowhere. And so standardizing is also a big part, which increases the value of the data a lot. And there were solutions to this before the pandemic. There are databases where you can upload these sequences. It's just that there is nothing there's no software available which you can just spin up and use as your own database and configure it as you like. There are kind of big players that have grown over 20, 30 years, and they've custom built things. But at this point, there's nothing that just, you just spin it up like this. Oh, because we have a new pandemic, we're interested in sharing, this virus X, and it has these specific requirements. We can just do it like that. There's nothing that exists at this point. So this is kind of one angle of the problem we're trying to solve. So I've mentioned this left part. While there are solutions, there is no open source piece of software you can just buy as a genomics database for this use case, pathogen genomic sequencing. The existing solutions, they've also got issues with, because the amount of data that was shared wasn't so high, there was no need for kind of good programmatic access. With the pandemic, things changed a lot. The interest went up a lot. People wanted to develop dashboards that were live and so on. So, and this was kind of rough. And so as users and producers of the data, we felt a need that there needs to be a better way. We can do things better now in the sort of in the year 2024. We've learned how to do things on the web. Let's do it in a better way. And while you can kind of put things into, say, Postgres and write a little web API around that, REST API, it's kind of the genomic, like everything in biology is always really complex. You come in from the outside, you think, oh, it's just a string. But in fact, the devil is in the detail. Biology is incredibly complex, and so if there are a lot of kind of special cases you need to handle, and so you can't just put the data in a database. It's not gonna work. Especially if you get data from hundreds or thousands of people around the world, you really need to validate it. And so there's a need to have something that's really purpose-built. Otherwise, it won't work. And so that's kind of the summary of kind of the idea of what we're trying to build. It's a tool that allows labs, a single lab or a group of labs or a community to have this data sharing hub that serves as the middleman between the data producers and the data consumers. And we've set out on this journey, I think, a year ago, and Fabian will now take you through sort of the more technical details. In the end, I'll give a quick demo to show you that what we have works already, and yeah, then we'll finish off. So now I hand over to Fabian.
Yes, thanks. So you've seen this image. Okay, we're coming from the left side, somewhere patients get sick, then we sequence the samples, we get somehow data, and on the right side we want to do analysis. But we need to understand, okay, what is this data actually? Who has worked with biology after they left school? A few? No one? Okay. So you probably also don't know what this genomic data actually is. So let's look at this. Maybe you still know the picture on the left side. It's an image of an RNA sequence. You got those little sticks in different colors. They are different bases. So that's basically what we put in a sequencer. And what we get out are fragments of the sequence, and then we can combine it to a whole sequence. And that's basically what we see on the right side. So in this example, we have 5 sequences consisting of letters. So it's a T, A, G, and a C, standing for the different bases. And just to give you a hint of the size of the data that we're dealing with, for SARS-CoV-2, this is around 30,000 characters long. For monkeypox, or mpox as it's called now, it's around 200,000 characters. So this is already 200 kilobytes, just one single string. And now imagine we have millions of those. That's huge data sets. So, but as it is, it's just a bunch of strings. How do we make sense of this? We need to align it. So how does alignment work? First, we need a reference. It's usually a sequence defined by scientists, more or less arbitrary, but of course, they know what they're doing, so they define kind of a prototypical version of the virus and a genome for that. And then we can align the sequences. There are algorithms that do that and kind of match the individual sequence to the reference. And then it's easy to identify mutations. So mutations might be substitutions, as we see here. So in the reference, there's a G. Sequence 1 has a T there. That's a substitution. We might also have deletions. As you can see in sequence 5, we denote those by a dash. We might also have insertions where the sequences have something more than the reference, but we don't see this in this table because we cannot really display it here. But that's one part. Once we got the alignment, it's easy to identify mutations. Next, we can translate it to amino acids. So also kind of how biology works, how our bodies work, is they translate DNA and RNA to proteins by looking at the sequences in triplets, and somewhere in the sequence there's a start codon, and then it translates the following triples to amino acids. The amino acids then combine to a protein which takes a specific form and then, does the job in the body that it's supposed to do. In the example of SARS-CoV-2, there's one nucleotide sequence aligned and unaligned, and also there are 12 amino acid sequences, one for each protein that the virus has. Okay, so this is one part of the story. The other part is metadata. So every sequence has a bunch of metadata. One's kind of true metadata and the other one is derived. So the true metadata might be, for example, geographic information. Where did the patient come from? How old was the patient? Those kinds of information. And the other one is derived from the sequence itself. There's lineage information that also Cornelius showed before. most well-known probably the WHO clades, which might be Omicron or Delta, or there are more specific ones like for COVID, we have pango lineages. Also, there's quality data, so basically saying how well the sequence was sequenced by the sequencer, so how well the sequencer did its job. That's kind of the data we're dealing with. Now we can think about what we actually want to do with that data. So the platform that we're trying to build first should support uploading the data. So suppose you have a scientist working in a lab, getting the data from the sequencer, and that scientist should be able to upload the data to the platform and then also manage it. Managing means, for example, you notice, I don't know, maybe you made a typo in some metadata, or you resampled the sequence and got a better result. So you might want to fix that. The next step is pre-processing, which mostly does the steps that I just showed you, to enrich the data so that it becomes more useful, so that in the next step, we can query the data. For example, saying, okay, hey, Loculus, give me all sequences from the UK, or all sequences of a certain lineage. And then as a user, you want to query that and also download the data, either by just clicking on a button and getting the sequence files, or via an API, getting the data and showing it in graphs. Next, let's look at how we implemented all of that. So that's an architectural overview of kind of what we built for Loculus. At the top, you see the query engine in orange. That's a separate project that we reused here. In blue, we have the Loculus code. So first, the website, the backend, and the pre-processing pipelines. The website was built in TypeScript and Astro. Maybe you've heard of it. It's a server-side rendering framework. So basically serving static HTML, but with little reactive islands that we wrote in React. The backend was basically built in a base decision in Kotlin and Spring, which worked quite well. The whole thing is backed in with a Postgres database where we store the data, and also there's a keycloak involved for handling, for doing the login. Let's look at it in a bit more detail. First, the query engine. How do we actually get the data out of the system? As I said, this is a separate project, consists of two components, Lapis and Silo. And this can also be operated standalone. So it's already published. It's not fully developed. We're still kind of adding a couple of features, but we're already using it outside of Loculus. So it consists of two components, as I said, Silo, which is written in C++, and is a query engine optimized for this genomic data. So for example, you could send queries to it. I put one there in natural language, get all sequences from the UK that have a character A at position 500 in the genome. So what does that actually mean? Get all sequences just tells Silo, okay, just give me all the metadata that you have, filtered by something, and the by something is from the UK, so that's more or less obvious. And then that have a certain mutation, which is an A at position 500 in the genome. So that's kind of an AND conjunction of sequences from the UK and a certain mutation. And then we built Lapis around it, which is a convenience API, also written in Kotlin and Spring, which mostly does mapping and a bit of convenience so that you can operate Silo from a browser more conveniently. Let's look at what this works. Don't look at the details. This is meant to overwhelm you. So let's instead focus on the red box. This is a really huge string. Actually, I've shortened it so that it fits in one line. And this is basically a query that we can send to Silo. And it's just a couple of mutations, so this mutation and this mutation and so on. And we've been using this in a dashboard that we built. And so you can do this live on the internet and get a couple of graphs within seconds. This sounds basic, but it's actually quite impressive because the underlying data is 500 gigabytes of genomic data in the raw form. If we kind of process it so that we can import it in Silo, it's around 1 terabyte of data. The data consists of 60 million rows. That's how many samples we have for COVID. And in Silo, we have around 100,000 columns. So it's actually quite impressive that we can do this in real time in seconds. Let's look at Silo, because it's actually quite interesting how we achieved that. As I said, it's written in C++. I always think of it as an in-memory database that consists of a single huge table. So if you look at the top right, there's this table of sequences. It's basically what you've seen already before. It's the aligned sequences. And now we can transform it to the bottom representation. So we simply ask, is the nucleotide a T? And of course, we have to do this for every nucleotide, also for G and A and so on. But let's focus on T for now. And then we just have ones and zeros, which are essentially bitmaps. And now we can store this in columns. And that's basically the whole trick. And now if you, for example, query for the data that says, OK, give me all sequences that have a T at position 2 and a T at position 3, we just take the two bitmaps, which is a lookup, which is simple to do, and now say, OK, let's concatenate them with an and operations on bitmaps are trivial. So basically, the filtering step is very, very fast. That's basically the whole trick. And now we use roaring bitmaps, which have an internal compression algorithm. And then we get compression ratios so that we can actually fit the whole data set in RAM, in memory, in around 60 gigabytes, which is also quite impressive because, as I said, the original data set is 500 gigabytes. The data set that we actually import, preprocessed, is around 1 terabyte. So this is a compression ratio of 95% roughly, so that we can actually almost run it on a laptop. And we export that as an HTTP interface so that Lapis can talk to Silo, and then we can operate it from a browser. So that's one component. The other component is the submission. So how do we actually get data into the system? And that's mostly the Loculus code consists of code for that. So it's the website, then we have the backend, which orchestrates all of that. And yeah, let's look at it in a bit more detail, how it works. So what kind of the lab scientists do is they upload files. It's a TSV file that's essentially the metadata table, and also FASTA files, which are kind of a text representation of the sequences themselves. So you upload a metadata file and a sequence file and submit that to a Loculus instance. The Loculus instance, so the backend will accept it, store it in a Postgres database. There's no real validation yet. That's what the preprocessing pipeline is going to do next, and also a couple more things that I'll tell you on the next slide about. And yeah, after the preprocessing pipeline is done, the backend will validate whether the data is complete enough to be imported in the query engine. And that's then kind of the final step of the submission process. So what does the preprocessing actually do? First, it does the alignment. So basically the table of one of the first slides that I've shown you. That's the job of the preprocessing pipeline. Then it will also translate it to amino acids. So basically, it's not just the raw nucleotide sequence that we import, but also the amino acid sequences. It will compute the derived metadata, probably most important, the lineages. It will validate, for example, checking. You might, for example, check, is the country valid? So I don't know, did they spell it correctly? Did they write just UK there and it should be United Kingdom? For example, is metadata present at all? Are sequences present? That kind of stuff. If something's wrong, the pipeline will report errors, blocking sequences from being released, or issuing warnings telling the submitter, hey, this looks suspicious, maybe you want to check it again. So I've understood what we kind of want to achieve. Now everyone should be able to host that thing. So we want to make the deployment as easy as possible, because it's actually quite a complicated application. You need two external things, which is a keycloak and a Postgres database. And then the Loculus internal part is configuration files, because we have a bunch of applications that you just saw. Each of it needs a configuration file because you need to configure which organisms should actually be available in the instance. You need to configure which metadata is available, what the reference genome looks like, those sorts of things. And also you need to kind of supply a preprocessing pipeline, either one by yourself, or you configure one of those that we provide that goes to the individual artifacts. And then everything is available as a Docker image, and so you basically just need to launch a couple of images, have containers running, and that's basically it. To simplify the whole process, we provide a Helm chart, because on the one side, you get a Kubernetes deployment for free. On the other side, it simplifies kind of wiring it all together. So we just yes, we provide one configuration file to the Helm chart, and then that does all the rest, putting it into the individual containers. Okay, I think next up, Cornelius will take over and give you a little live demonstration of what we've built so far.
Oops. Yes, so here's a demo instance that we kind of created in a couple of seconds, but just editing a line and making a new PR, because deployments now are really that easy for us, which is one of the goals we wanted to achieve. So you see here kind of the landing page in an actual production setting. You'd make it prettier, but this is sort of just the bare bones to show you how it works. And I can select the virus I'm interested in this case, mpox virus. It used to be called monkeypox. There was a big outbreak 2 years ago. Maybe you've seen it in the news. So there's also a lot of data available there. We've already ingested it all from the publicly available data to have something to play with. You see, for example, here this is the view for the data users. They can, for example, very quickly filter down by a country. If I want to look at all the sequences that were sequenced in Australia, it works really quickly. I can also do querying by lineage. There are different lineages in clade similar to COVID. I can select, for example, lineage B1, and again, I now have only 64 sequences left. I can also do some sort of querying on the length and so on. So these are the things that the databases that pre-existed, they sort of support. Maybe they're a bit slower because technologically they are not they were developed a bit in the past. And so this is kind of the viewing side. There's also for each sequence a detailed view. So if we click on, for example, this sequence here, we try to present all the data we have in a way that is attractive. We also try to cross-link things. You can see here the substitution. So these dashboards, they tend to be a bit overwhelming to kind of uninitiated users because this is software for scientists who are used to kind of looking at very complex stuff. So in many ways, that's a challenge, how to present that complex information in a way that it's digestible by the scientists. And so I can show you quickly, for example, how quick one of these mutation queries is. You can see here, let's copy this mutation here. Let's go back, put it in here. Oops, why does the pasting not work? Well, I've remembered it, I think. Yep, and there it is. And so now we've quickly filtered down all the global mpox sequences by that mutation. And so that's something that people would do in practice, quickly looking up which sequences contain which mutations. So that's the view component. And then the other very important component is, of course, the submission. Just to demo how this works, so you click on submit, and then all you need to do is upload a sequence file which contains these sequences and a metadata file. I can show you quickly how they look like, just so you get an idea. I mean, the sequences, they're just strings, as Fabian said, long strings. I mean, it just keeps on going, right? And the metadata, it's just boring stuff. And I can just upload them here by clicking. Oops. Well, let's use the right ones that I've prepared. Drop the metadata here, drop the sequences here, and submit. So one key thing here is that's kind of one of the innovations we're trying to achieve, is that the analysis that just happened, you saw it spinning, that happens almost instantly. The usual processes of the existing databases, there's often manual steps involved, so it will take a day or two, sometimes even a week. Here we've managed to do that kind of immediately, which is really good, because then people who upload, they can immediately see feedback, they can fix things if we provide warnings. They can immediately make a decision, and they've just got it out of their mind. You can also edit. If I spot there's a problem, I have December here instead of November, I just change it, resubmit, it will do the validation, and here we've got the results. Oh, not yet. Maybe I crashed it. Oh no, here it is. yeah, you can also kind of delete your sequences and affirm that they should be released very quickly. And yeah, then 30 seconds later, it should appear in the browse view. We've also, of course, so that's another thing we try to do, kind of have good documentation, so you can do programmatic submission. We've got Swagger for that, which details all the endpoints, how to upload data. It's really quite straightforward, simple, post HTTP call, and then you've got the data in the database. Now that doesn't sound, revolutionary, but if you're a bioinformatician who's been fighting to submit data, it's often taking a lot of time to do that, and this is quite new to the space. I mean, bioinformatics has kind of a tendency to be kind of a little bit outdated. People still use lots of Perl scripts and things. It's not as kind of modern as web development in many ways. So now if we go back, we should be able to see the results. No, they haven't come through yet. Okay, then maybe I can show them at the end, once I give back to Fabian. It should have worked, but apparently it hasn't yet. Okay, Fabian.
Please.
Yeah, you can. I give back to you.
Okay, yeah. So I hope you've got an impression how kind of the problem and the solution we're trying to achieve. And actually, in my impression, it works quite well. We hope that actually people will use it. So we've been developing this over the past year. So we started exactly a year ago in July last year. That's the commit history. So there's been a little Christmas break, but otherwise there's been continuous development. It will also continue for a bit still. So we plan to release the whole thing as an open source software. It's not public yet, but as soon as we feel ready, we will publish it as open source. And that is hopefully going to happen in the next couple months, but pretty much definitely this year. So yeah, we'll see on how that goes. We would like to add a couple of more features in the future, because this is kind of the minimal thing that we want to operate with, a couple of pitch features. One thing that we want to add is sharing raw data. So actually what you get from a sequencer are sequence fragments and not the whole sequence. And we want to enable people to also share those for additional analysis. We have no idea yet how we want to analyze that, but that's an open question that we're trying to solve. We also want to enable crowd curation, which means, for example, someone uploads data, a user spots that there's a mistake, then they should be able to first, mark the error and also suggest corrections. So saying, okay, the sequence is actually, I don't know, from Africa and not from Europe. And another thing is federation. So as we said, the target is that kind of every lab, every country, every university can host their own instance. So we would like to make them communicate with each other. So that, for example, you have labs in Switzerland, and then you have a Swiss instance for whole Switzerland, and every lab in Switzerland can then kind of have their own instance, submit data there, and so that it, for example, automatically syncs to the whole Swiss instance. So basically, we're at the end now. Maybe a bit of a private opinion on how the project went. So it was a software development in scientific area, which was at least new to me. So most of people the people at TNG come from science somehow, but usually they don't really work in kind of scientific software anymore, which was quite fun because it was a very small and very agile environment. We were just a couple of people involved, so it was very fast and very, very agile actually. We were using Astro, so that was also quite a fun experience because it kind of got you back into the old school HTML development, because you send a request to a server and you get back an HTML document, which works quite differently from how nowadays most front-end applications work. We try to develop open source software which has its own challenges, because you want people to use it, people that you've never heard of before, and also you want to have other people develop in the code. So you need to kind of document it, make it understandable. We have quite many languages and frameworks, which is actually nice to learn, but also has its challenges, because, I mean, if you change the artifact within the same repository, you get a totally complete different language or framework. Also, we tried to make everything configurable, which is by itself hard, because you cannot just, write the code, but you always need to look at the config, and So it's kind of indirect coding because you always need to kind of check the config and then generate the HTML and so on. We used Helm to help us with that, but that also has its downsides because it's quite powerful on the one side. On the other side, if something goes wrong, there's quite bad tooling to investigate it. Maybe also because we misused a bit. But actually, we also feel the benefits. But yeah, if something goes wrong, it's hard to investigate what kind of you did wrong in the initial config that you provide to the Helm template. And also we used ArgoCD, which was incredibly useful. Also the live demo that we just showed you was essentially tweaking the instance a bit that we have for our main deployment and creating a pull request. That's it. We have an instance set up. And that works for basically all pull requests, which is incredibly useful because someone develops it and the other one can just go to the browser and investigate. You don't need to check out the code locally. Also kind of less code people can also just check it, go to a pull request and see, okay, that feature actually looks nice. Who actually developed it? It was a wild collaboration of international partners, also quite fun to work with so many people. Also we as TNG were hired to support externally on that, which was actually a nice collaboration. So now we're at the end. Thanks for the attention, and yeah, happy to have your questions.
Great, thanks for the talk, and we already have questions.
So I'm curious, what is approximately the total number of people that were involved in developing this?
So what was the total number of people?
How many people were working on this? And the other question is regarding the technology you picked. You have Kotlin, C++, Spring. Who made the decision what languages to use and so forth?
Okay, how many people? It was different, 5 to 10, so that was kind of the rough size. And also, that's it. So it's not kind of that's the development team and then you have the business department behind it. No, that's the team. That was kind of the small team size that I mentioned.
Yeah, what's quite special, I guess, from a software development perspective is that, I mean, the people from the scientific side, they are also developing. And we kind of dog fooding our own thing here because we would be the eventual users. So that's something that maybe speeds up the feedback loops quite a bit. Yeah.
Concerning technologies, it was basically a decision based on the problems. So, I mean, we want to design a web API. What do you take for that? Well, Spring is the first decision that you usually want to take. And it also worked very well because it's just reliable and it works. Then you want a highly efficient database. So what do? You pick C++. Then you want to build a front end. Don't do that in Spring usually nowadays. So you pick something in TypeScript.
I just wanted to show that the data is now available. This is what we uploaded. You can see here that today is the 5th of July and 10 a.m. UTC is 12 p.m. our time zone. Yeah, so it works.
We have a question from Zoom, if I can read that out.
First take the Zoom question.
So I'll read it as it's written. Can you explain a bit about what raw data is and why it would be good to have this included too? As you already have sequences which you go to a lot of trouble to align and compress, what would this add?
Sorry, I'm not quite sure I understood the question. Can you repeat it?
I can repeat it, yeah. Can you explain a bit about what raw data is and why it would be good to have this included too? As you already have sequences which you go to a lot of trouble to align and compress, what would this add?
Okay, yeah, the question is about raw data. Why should we look at raw data? So, because biology is so complex and the technologies involved are so complex, there is a lot of things that can go wrong when you turn that raw data into that kind of simple string. And there's, in a way, it's kind of a statistical problem, and it's very easy to make mistakes there if you have some config wrong and so on. And this happens all the time. That's why QC is so important as well. I mean, basically, almost every second sequence, if you look at it, you can tell that something went wrong there in the automatic step as a human. I mean, and QC can also do its share. So because you have also so many labs producing it, and they all use their own pipelines to turn that raw data into these output assembled sequences, there's a benefit to having the raw data from all the labs in one place, and then you can run one single pipeline on that to have consistency. Yeah, when something crazy comes up, often what people want to do is they want to see the raw data to see whether they trust it, or maybe it's an artifact of the analyses. And that has happened a couple of times. I mean, there were a couple of scare recombinants I remember in the news, maybe you too. It turns out the simple string, you can't tell whether it was done correctly or not. If you look at the raw data, it was clear that it was just maybe a mistake in the way the sequence was assembled.
So maybe in simple words, imagine you have a string that's 1,000 characters long, you know what it should look like, and now you get pieces of 10 characters, and a million of those, and now you need to kind of puzzle it together to the original string. That's a hard problem, and a lot can go wrong. And the other thing is maybe one question that you can answer is, for example, also if you have a patient that's actually infected by 2 variants, you won't see that if you combine it into a single string, because you might actually need to combine it into 2 strings.
So thank you. And do you also envision to extend the current platform to support the other type of data?
To support what, sorry?
To support other type of data.
What kind of?
So for example, right now, this is the genomic data, right? So there could be also in the different industry, for example, like patient data, customer data, etc. So is it something that you envision?
Not really. I think. I mean, if you think that this type of kind of specialized solution works for something else, I'd be surprised. Welcome to hear. I think a lot of the value here is in kind of on purpose narrowing your scope and making a custom-built solution. So there are already general things, for all sorts of bacterial data, human genomics, and so on. And that's one of the problems of these existing databases, that they need to cater to all these use cases which have different requirements. And so by saying we only look at mostly viruses, I mean, you can also put bacterial data in there, but our background is mostly viruses. If you put, but that kind of helps us to simplify the problem, and we can, yeah.
So my first question was very similar. What's the reason for limiting yourself to viral data? And maybe the second one is, so you have all these big players like GenBank or like European databases. So in bioinformatics, one of the main things you need is all the data available, kind of. So you need to go to these big databases that have all the data available. How is Loculus kind of, which gap is it filling in a way?
Yeah, so remind me quickly of your first question. I just forgot.
I think you answered that already. What's the reason for limiting yourself to viral
Oh, yeah,
data?
okay. So that's simple, because bacteria, the bacterial genomes are bigger and they're much more complex. And so it's much more difficult to, you can't just call mutations. They might not be the relevant entity to look at. And so because of the increased complexity of for example,
the
bacteria.
pre-processing steps, because the rest is similar.
Yes, and also there's kind of, people sequence fewer bacteria, and they are all just each quite different to each other. Viruses are really neat because they're just, these strings of 30,000 or so, or 10,000 KB.
And then there's mammals, there's,
Yeah, exactly.
Archaea,
yeah, and so the virus simplicity really kind of is something that makes a lot of these things possible and easier. Your second question was?
Like big databases?
Oh, the big database, yes. So, in fact, I mean, what we've got here is all the data from the big databases already. I mean, we've downloaded the data from there and added it in here. And so there's not, if you do an analysis on mpox now, for example, if I'd submitted these fresh, right, you'd have now everything from the big databases plus what someone submitted here. And so,
but why would I want to do that if it's already available in the big database?
Why would you? Oh, certain things you can't do on the big databases. So first of all, I mean, sometimes downloading things can take a long time. They might not support the queries. Like a lot of the dashboards that Fabian showed, you can't do them out of the box with those big databases. And one reason to do it is to simplify the submission. The big databases, if you want to submit there as a submitter, it takes a lot of work and time. They often don't have any automation. You need to send emails, put something on FTP, and learn the file formats. And so having a simpler way of submitting is very valuable. And it's not difficult to go from here. We're building right now something, and that was maybe missing, that will submit to the big database. So we kind of take that away from people. They only need to submit to one simple place, and then the final submission is.
That was a missing link. I think,
yeah.
That you kind of connect your data to
right.
others.
Yeah.
In a way.
There's another aspect. Like there was the mpox outbreak like 2 years ago, I think. Maybe you've heard that in the news. Suddenly there was kind of, they called it monkeypox back then, now we call it mpox. There was an outbreak somewhere in Germany and in other countries. Imagine that happens again with a new virus. You can spin that thing up like in minutes and have a new instance for a new virus. And that's kind of the thing that we're also trying to tackle. So if there's a new pandemic, we hope there's not, but if it comes up, or just, I don't know, local outbreaks might also be sufficient. Then you can spin up a Loculus instance tailored to that virus, and that's it.
Yeah, and it can easily take days. I mean, these even if they fast track, you know, it'll take a day or 2 or 3 for something to show up. And so what people do instead, they post it on some discourse forum where they discuss it. That's really what people do. And so then, as soon as you have multiple people sharing it to discourse, there'll be someone else who downloads it from discourse and puts it in their own file on GitHub. But kind of all this manual stuff you can just solve if you do it like that. I mean, the benefit, if you don't care a lot about kind of the analytics powers and the kind of really short turnaround, then it might not matter so much. But when Omicron hit, for example, BA1, everything happened so quickly. You know, people were making decisions on whether to shut the airports or not. Controversial decision, but you know, having better data never harms. And so, yeah.
Sorry, one quick comment, Fabian. There will be a next pandemic, number 1. 2, who funded the project and why? And last question, data security.
So data security, that we have almost no personally identifiable information. So I mean, we've been doing our best. We've done a penetration test and so on to check that everything is safe there. So I think that we're taking care of. Then there was the question, who funded it and why?
Who funded
I mean,
and why?
one of the I was funded. I'm not kind of at the PI level in charge of the funding stuff. I think that, you know, professors have some sort of money they can use as they see fit, and so that there was no grant yet that is funding this. It's kind of, you know, people also working in a way in their spare time on the side, you know, besides their normal jobs, doing this, which doesn't scale maybe, but
Okay, great.